The overall goal of the following experiments is to specifically label DNA and proteins using methyltransferases, which naturally transfer the activated methyl group from the cofactor S-adenosyl L-methionine, called ADOMET, to DNA, RNA, proteins, or small biomolecules. One-step labeling is achieved by replacing the natural cofactor ADOMET with synthetic aziridine cofactors that change the course of the reaction, leading to enzymatic coupling of the whole cofactor and therefore covalent labeling of the substrate. Sequence-specific methyltransferase-induced labeling of DNA is demonstrated with the adenine-specific DNA methyltransferase MBSE-C1, which couples the biotinylated aziridine cofactor 6BAZ to the AGC-GAT recognition sequence, leading to sequence-specifically biotinylated DNA. Methyltransferase-directed transfer of activated groups is used to label protein, and is demonstrated using MTA's set79, which transfers a propargyl group from C-adoine to lysine 4 of histone H3. The alkynylated lysine residue is chemically labeled by an azide-modified fluorophore using copper-catalyzed click chemistry, thus leading to specifically labeled protein.
An advantage of using methyltransferases for labeling is that they have the potential to directly target native substrates. I will demonstrate this by sequence-specific biotinylation of plasmid DNA.
Methyltransferase catalyzed protein modification with the cofactor analog Z-adenine enables the introduction of a wide variety of reporter groups in 2 steps. I will demonstrate this by fluorescence labeling of the protein